Hello, my name is Taye, and this is my project, Novel Methods for the Continuous Measurement of Cell Vibrations in Real Time using Optical Tweezers. Living cells are complex systems that experience morphological changes and exhibit motion as a result of their biological processes. One of these processes, cell-to-cell adhesion, is shown in the video below. Here, these hair-like appendages responsible for cell adhesion result in tremendous movements of the cell. And previous work in the Omering lab has demonstrated that at least to some degree, upon recording the spectrums of vibrations produced by different cells, different cell lines, cancerous versus non-cancerous, can be differentiated by their unique vibrational patterns. But this research was only able to prove so at discrete points in time. So, the objective of this research was to precisely and continuously record the vibrations of cells to analyze how they're impacted by treatments that kill them. To do so, we utilized optical tweezers, which utilize optics or high-intensity light to grab different cells with no physical contact, only the effect of light. To do so, this relies on the principle that light carries momentum. So, if we have a microscopic particle like a cell here under the trap of the optical tweezers, when high-intensity light comes in and refracts to the right, it produces an equal and opposite reaction on the cell, pushing it back to the left. Though the same is true on the far end of the cell, pushing it slightly back to the right, the net movement will always be towards the center of the trap so long as the intensity there is greatest. And the same is true in the vertical direction, where the cell will be pushed up or down to the center of the trap with no physical touching, which minimizes environmental disturbances and allows the cell to vibrate freely. So, to summarize the methods for this experiment, we trapped highly active U251 cells with optical tweezers and then treated them with paraformaldehyde, which is a fixed-ebb agent that cross-links and covalently bonds the proteins in the cells together to arrest all movement and freeze them in place. We utilized a cell tram, which, as seen here, is a highly precise syringe that administers small microliters of treatment to minimize all environmental disturbances. And throughout this whole process, we recorded vibrations before using bioinformatics to statistically compare the oscillatory patterns of the cell before, during, and post-treatment. Visual analysis throughout this experiment showed to us that following treatment, cells would produce unique blebbing resulting from their apoptotic mechanisms engaging before the cell was fixed in place by the paraformaldehyde. This proves to us that cells treated with paraformaldehyde were in fact treated and produced graphs like as follows. As can be seen here, in this real-time graph of the force produced by the cells over time, in the first 47 seconds, we have the baseline vibrations of the cells that are then disrupted by the treatment of administration of PFA by the cell tram. However, following treatment at 390 seconds, the vibrations return back to their normal levels. Interestingly though, our research showed to us that these vibrations taper off in intensity over time, and by the end of the treatment, the standard deviation of the vibrations from their mean levels are reduced by 3.8 times from the initial levels, which statistically proves to us that paraformaldehyde reduces the intensity and amplitude of cell vibrations with time. Another qualitative graph here shows that just immediately following treatment, the vibrations of the cell are still very thick in amplitude, but as time goes on, these vibrations taper off, showing that the cell has become less active with time. Now, so far, we've only demonstrated a correlational relationship between paraformaldehyde and reduction in vibration. So in future experiments, we'd like to causally solidify that causal relationship to prove that cellular activity itself is being reduced and attenuated by paraformaldehyde. Ultimately, This novel method, showing how cells are immediately impacted by treatments, has the possibility to significantly improve upon current chemotherapeutic treatments, which sometimes take weeks to months to show traditional changes in phenotype. Thank you for watching and listening to my presentation.